This is Dr. James Hildreth, a very prominent researcher and academic physician in the field of virology and HIV research. He's currently the president and CEO of Meharry Medical College, but he was a full professor at Johns Hopkins, and he wrote this paper with two of his colleagues there. And what he said, and I quote, "The virus is fully an exosome in every sense of the word."
Cells by nature work to heal, repair, and reconstruct the body. A growth factor is naturally occurring protein capable of stimulating cellular growth, proliferation, healing, and cellular differentiation. Usually it is a protein or a steroid hormone. Growth factors are important for regulating a variety of cellular processes. Let's find out how exosomes are made. Inside the cell, a cell membrane forms inward, forming a special type of lipid vesicle called an endosome. Then the proteins, microRNA and messenger RNA, invaginate the endosome's membrane, forming the exosomes. Endosomes then fuse with the surface of the cell, releasing the exosomes outside of the cell. Due to the observation of this process, historically exosomes were believed to be a waste byproduct of cells. But when researched further, and it was discovered that, in fact, these are the mediating mechanism involved in direct cell-to-cell communication. These produce an anti-inflammatory response due to the communication from the many proteins that down-regulate inflammatory proteins. Other proteins will then recruit stem cells to the area that will help unregulate these cells to proliferate and promote organized healing through the regulated process. Then once the task is complete, there are additional proteins that tell the cells when the healing has been completed.
Exosomes. And I'm going to talk about what they are and why they're relevant for this discussion. So exosomes are something that is naturally occurring in the body. So I have this diagram here, and if you look at the top right of the diagram, you'll see that this is a normal cell here outlined, and inside the cell you have these vesicles. And inside of our cells there are a number of different little organs, they call them organelles, and they're generally contained by a membrane. They come in different shapes, but lots of them are in this structure. It's essentially like a spherical blob that has the same membrane as is the outer membrane of the cell, a lipid bilayer membrane, and it can contain various types of chemicals. Now, this is a specific type of vesicle that will end up merging with the cell membrane at the surface under certain conditions and release these exosomes out into the extracellular fluid, and they'll get into the circulation and be distributed around the body. So while they're inside the cell, they're called MVEs, which stands for multivesicular endosomes. So in other words, they have a bunch of exosomes or vesicles inside them. And they release these on a regular basis, just day to day. And there are many things, though, that can induce this process and accelerate it and increase the number of exosomes that it releases outside of the cell. And so these exosomes leave the cell, and you see they have these little squares on them. And these little squares are like a lock. And what they do is they go around the body through the circulation, and they're looking for the right key to fit their lock. And that's called the target cell. And depending on what kind of cell releases them, they might have different keys and different locks, so they're targeted to different parts of the body. And these are are mostly thought to involve communication. So communication between one cell and another cell, between one part of the body and another part of the body. And they can have many functions in this communication, which I'll get to momentarily. So here on the left, this is an electron micrograph, or a picture from an electron microscope of exosomes. And you can see here an exosome budding out of the cell. And it's essentially a spherical or circular, this is a cross section, so this is like a slice of the tissue, where on the periphery there are like these kind of globular densities or little dots or circles, right? And inside the cell here where it says MVB, sorry, these are the same as MVEs, that's where all these exosomes are when they're inside the cell before they bud out, okay? And now we have a picture on the right which allegedly shows this COVID-19 virus. And you can see that there are these vesicles budding out of the cell in a circular shape with these globular dots on the periphery. So essentially it's the same thing. Now you might think that this looks a little bit fuzzier on the exosomes and it looks a bit sharper in this picture. And I want to tell you that the reason for that is because when you're cutting these thin sections of tissue to make these slides, you're using this device called a microtome. It's like a vibrating razor blade. And it's technically difficult, and sometimes the tissue doesn't cooperate well, or sometimes the person is less skilled, and you might not have a perfect slice. On this COVID-19 slide, this is, I've not seen a better slice than this. It's absolutely perfect. The one on the left is a little bit thicker. And so that's why it looks a bit fuzzier and less sharp, but essentially you're seeing the same exact thing. So I happen to be looking in the virology literature, and actually they also think exosomes and viruses are possibly the same thing. So this is Dr. James Hildreth, a very prominent researcher and academic physician in the field of virology and HIV research. He's currently the president and CEO of Meharry Medical College, but he was a full professor at Johns Hopkins, and he wrote this paper with two of his colleagues there. And what he said, and I quote, "The virus is fully an exosome in every sense of the word."